I'm really excited to share some interesting results I got last year from the study that I'll be talking about today. So to introduce myself, my name is Carter Pilkey. So before I get into my personal research, I'd like to give a little bit of background on the research lab that I am a part of in case you are a newbie to the meeting or if you're unfamiliar with our research that we do. I am part of the American Chestnut Research and Restoration Project, which is based at SUNY-ESF, which stands for Environmental Science and Forestry. We are in Syracuse, New York. We are a nonprofit program that has been in partnership with the American Chestnut Foundation since 1990. We are directed by Dr. Bill Powell, and this is the rest of our research team listed right here that's currently working for the project. So we're a pretty big team, but this research has also been a culmination of many graduates and undergrads and volunteers and collaborators at different universities that have contributed to this research over the past 30 years and have gotten it to where it is. Our lab at ESF focuses on researching the American chestnut tree and figuring out ways to infer blight resistance to the tree, which was essentially decimated by chestnut blight, as we all know. And our method of inferring blight resistance was through genetic engineering, in which a gene called Oxalate Oxidase, or OXO for short, which was cloned from the wheat plant, was inserted into the genome of the American chestnut tree, which gives it its blight resistance. You can see here from some small stem assays that were done with the American chestnut trees that have the OXO gene that was inserted. The central trees are surviving the infection 6 weeks after inoculation in comparison to the wild type American chestnut trees on the left, which have pretty much all died. And also they're tolerating the fungus's infection even better than the Chinese chestnuts, which is shown to the right here. And here's a close-up shot of the infection site. So the tree to the right, which does not have the OXO gene, just a regular American chestnut, really nasty infection in comparison to the tree on the left, which has the OXO gene for defense against the blight, which it is protected. So we do a lot of studying of the Castanea species. And so we're really interested in kind of using the tools that we have to research these trees and start applying them to other species of trees, one of which includes the Ozark Chinquapin. The compounds that I'll be talking about today in my research is known as Oxalic Acid. This is a really common compound that's found in many different plant species, super important for calcium regulation. Plants also use this as a defense mechanism against herbivory. So us humans are also considered herbivores, so we can sometimes feel the effects of foods that are high in oxalic acid. These foods are considered being high in oxalate, as you might have heard before. The most common example is spinach. That's why after you eat spinach, you can sometimes detect that bitter aftertaste. That's actually the oxalic acid present in the plant. There's a variety of other plants that are also high in oxalates, including like rhubarb and kale. Sometimes people are put on low oxalate diets because the effects of oxalic acid can be detrimental to someone who might have like a kidney issue. This is because oxalates are really good at absorbing calcium. So if you have kidney function problems, eating these foods can cause stones to occur, which could be a problem. But for the average person, eating normal amounts of these foods is totally not a big deal and totally safe. Fortunately, some species of fungi also use oxalic acid to their advantage, and this includes Cryptonectria parasitica, which is the fungus that is responsible for chestnut blight. On trees that are susceptible to blight, the fungus will invade wounds on the tree and then will secrete oxalic acid in high volumes. This then lowers the pH of the chestnut bark and it kills the tissue where the acid was excreted. This fungus is considered a necrotroph, which means it kills its host and then feeds on the dead tissue, which is really nasty sounding and actually kind of perfect for Halloween today. And then the fungus will then eat the tissue that died, which creates those sunken cankers on chestnut trees. And these cankers will eventually girdle and kill the chestnut tree. In the Castanea genus, which includes all the species of chestnut and chinquapin trees, there is kind of like a range in blight resistance. We like to think of it on a spectrum. It's generally regarded in the literature that American chestnut has the least amount of resistance to the fungus, and no natural resistance has been found in the species. Chinquapins are regarded as having kind of an intermediate resistance to the fungus, including Ozark and Allegheny chinquapin. And then Chinese chestnut is generally thought of as having the most resistance to the fungus, the most natural resistance as a part of evolution with the fungus over millions of years. So what we're really interested at ESF is figuring out why the chinquapin species seem to have this natural resistance that their North American cousin, the American chestnut, does not have, and also what gives the Chinese chestnuts its higher resistance to this fungus. So we're really interested in investigating this research question further because we think it could help all the Castanea species as well as the American chestnut if we could figure out what is giving these trees their leg up against this disease. So to try to figure out which trees may have some natural resistance to the fungus and what could be giving them their resistance to the fungus, we like to perform a test called an exogenous oxalic acid leaf disc assay. And this is a pretty simple to conduct assay that helps us study individual trees by testing their leaves. So a little bit about this test. It is a method to quickly observe sensitivity to oxalic acid and possible blight resistance. So we expose the leaf tissue directly to oxalic acid, which was, as we talked about earlier, that compound that the fungus excretes to essentially kill the bark of the chestnut tree and then effectively kill the tree. So no blight fungus is present in this acid in comparison to the leaf assays that you guys at the OCF have performed. So it's quicker, a little bit more simple assay to perform. And it's also important to note that this assay was not developed by ESF. This method is used for studying oxalate oxidase in other plant species. So this test has been performed on soy, tomato, I think rice as well. So this was actually a method that we used to test our transgenic trees have the oxalate oxidase gene present in their genome to study them and gauge their resistance to the fungus. So as kind of our first investigation as to why the Ozark chinquapin could have some natural blight resistance, we wanted to start with this assay because of its simplicity and it gives us a good starting point to work from. We wanted to conduct one of these leaf assays with the Ozark chinquapin. So this is where my research began. It started with collecting leaves from tree individuals we wanted to test. So we collected a few leaves from 2 wild-type American chestnut trees as the no-resistant controls. So these should be really susceptible to the oxalic acid. We included one of our transgenic American chestnut controls that have the OXO gene. So we should see that they're protected from exposure to oxalic acid because the oxalate oxidase gene breaks down the acid and gives it its protection. We also included one Chinese chestnut tree as a resistant control. So this has, for some unknown reason, a natural resistance to the fungus that we're still trying to elucidate. And then also 4 Ozark chinquapin tree individuals, which is the resistance that we are trying to gauge. So kind of the focus of the study. And then we can compare all of these different leaf samples to each other and gauge their resistance to oxalic acid. So to make all the leaves equal sizes, we use one of these cork borers, and it acts kind of like a cookie cutter. So for each of the few leaf samples, we take an equal number of these cork borer discs. And then we make up a oxalic acid solution. So oxalic acid is actually sold at chemical companies in granulated forms. So we simply mix a specific amount of oxalic acid with water as our solution used in this test. So all of our leaf disc samples get put into their own little vial with an equal amount of the oxalic acid solution present, and they get put on a machine that rotates them so that all the tissue gets coated with the acid. And then after 20 hours, we removed all of the leaf discs and then grouped them together and then imaged them using a scanner. And then from there, we can go in with an image analysis software and then measure how much alive tissue is there compared to all the necrotic tissue on each leaf sample. So the necrotic tissue, here, let me pull up my little pointer tool. The necrotic tissue is this brown tissue that you can see here compared to the green parts that is alive. So right off the bat, we notice that these two wild type controls have much more necrotic tissue present compared to alive tissue. So that's a good sign. Once we see that the controls, the susceptible controls, have this largely necrosis surface area on their leaf samples, we know that the assay has worked. We then compare this to our other resistant controls, which include the Chinese chestnut. We can see actually there is a lot of green tissue still there. So that's good. That says, okay, we are seeing that resistant control, as well as our darling transgenic American chestnut also has a lot of green tissue left on their leaf discs in comparison to the wild type controls. And then of course, our samples in question over here are 4 Ozark Chinquapin samples is what we were looking at today. And this is where the exciting results come in. So after I measured all the leaf discs in the imaging software, averaged them, ran the stats, and graphed them to get our results. And right away, we can see our American chestnut controls had the least amount of alive tissue after being exposed to the acid. So essentially, they were killed the most by the acid, which is exactly what we'd expect to see. And now taking a look at our resistant controls, we can see the darling tree that has the OXO gene for blight defense has significantly more alive tissue than the wild type controls. And then the Chinese chestnut control also has significantly more alive tissue than the susceptible controls. And now for the exciting result, which is that these 3 Ozark Chinquapin trees, individuals appear to have tolerance to the fungus's deadly oxalic acid. They have significantly more alive tissue than susceptible American chestnut controls, so it's possible that this tolerance could translate to natural blight resistance. So just to explain this figure a little further based on the stats, any bars that share the same letter are not statistically significant from each other. So this Chinquapin here had the highest average alive tissue, but it wasn't statistically significant from the darling tree or the Chinese tree. So it has comparable resistance to these two because they share the same letter. And this 4th Ozark Chinquapin tree right here wasn't statistically different from the American chestnut controls, but it had comparable tolerance to the resistant controls, tolerance to the acid. So overall, these results are really interesting, and we're excited to keep exploring this tolerance. But lastly, I should say that this test is just one of the many tests that help determine resistance to blight. So this test doesn't necessarily prove the trees are resistant, but it definitely highlights the possibility of resistance and certainly highlights individual trees to keep investigating. Based on the results that we got from that figure, we wanted to know if Ozark Chinquapin was using the Oxalate Oxidase gene to tolerate this oxalic acid. And this is the gene, like I mentioned earlier, that the Darling 58 transgenic American chestnut tree has that gives it protection against the oxalic acid. So to figure this out, we do a really quick, simple assay. It's based on this chemical equation right here, where here is oxalic acid, our nasty compound. And here is the oxalate oxidase enzyme coming in, and it breaks down and neutralizes this deadly acid into carbon dioxide and hydrogen peroxide. So hydrogen peroxide is what is really important for this assay that we do. We use a solution, a histochemical solution, that can detect the presence of hydrogen peroxide by staining the tissue a dark blue color. So here's what the histochemical assay looks like. You can see here this top row of tubes. These have the histochemical assay, but no oxalic acid is present in these tubes. So it is a negative control. Where down here, the bottom tubes have the histochemical assay plus oxalic acid. So when we add a tissue that we want to test, if there is oxalic present there, if there's oxalate oxidase, the gene will then break down the oxalic acid and that hydrogen peroxide will be expressed. And the histochemical assay works by reacting with that hydrogen peroxide byproduct, and it stains that tissue dark blue. So just to quickly summarize, like here is the darling transgenic American chestnut tree, and you can see this dark staining right here, indicating that the oxalate oxidase gene, or OXO, is present. And you can see compared to just the regular American chestnut sample right here, that there's no staining occurring. So we put some of the Ozark Chinquapin tissues into this same assay, and thus there is no staining occurring, which means the OXO gene is not present, and it is not the reason for tolerance to oxalic acid. The million-dollar question that we now have is, where is this tolerance coming from? Like I mentioned, the Ozark Chinquapin does not use oxalate oxidase enzyme, or OXO for short, as its mechanism for tolerating oxalic acid. So could it be another enzyme that it's using to degrade oxalates and tolerate oxalic acid? There's a couple other pathways that have been researched and cited in the literature. One of them is called oxalate decarboxylase. This has been found in other plants and fungi species to tolerate oxalic acid. There's also another pathway that is more complicated. It is a 4-step degradation process that with many genes contributing. This is actually a hypothesis that we have for why Chinese chestnuts can tolerate oxalic acid. It's maybe using this complicated method, which is possibly part of the reason why the TACF has a hard time back crossbreeding and capturing all these genes that are necessary for being resistant to the fungus. Or is it a pathway or even an entirely different mechanism that hasn't been cited yet? So just to wrap everything up really quickly, the Ozark Chinquapin does appear to have tolerance to oxalic acid based on the leaf disc assays. It was a pretty small study, so we would like to repeat the study with more leaf samples just to corroborate the data and make it more robust. The tolerance is not a result of OXO or oxalate oxidase, but possibly from another oxalate degrading pathway or an entirely different mechanism that give the trees this protection to the deadly oxalic acid. If we were to repeat the leaf disc assays and we have the same results, we'd like to start molecular analyses on the Ozark Chinquapin to help determine any important genes that could be providing protection for the tree and giving it resistance. Because like I mentioned earlier, it could be really important for helping the other Castanea species that are susceptible to chestnut blight. And then lastly, this test could serve as another assessment of blight resistance in Ozark Chinquapin, along with the detached leaf assays that use the actual plugs of fungus to gauge resistance. So it's just another one of the tests that are important in getting a whole picture of each of these trees and their resistance to chestnut blight. I pulled up my little screen again here just so I could say goodbye to you guys, and thank you so much for allowing me to speak and listening to my presentation here. I know it can be a little bit overwhelming talking about chemical pathways and ACEs and enzymes and stuff. So if you have questions or you'd like to continue this discussion, please reach out to me. I love talking about this stuff. I love talking to other tree enthusiasts. So please shoot me an email if you would like to chat, or get in contact with Steve, and then he can contact me, or whatever method you think would work best. And thank you guys so much again. Have a great rest of your day, and happy Halloween. Bye-bye.
And Tori, I don't know, if I'm looking, I don't see anything yet on the questions, but if we do have them, we will go ahead and we will add those to it. And I can't say enough good things about what Hannah's doing. And again, this just kind of reconfirms what we've been doing. Interestingly, the leaf samples that she took the plug from were actually not from the parent trees. They were from open pollination on our research test plots. So what I sent to her, they went ahead and they germinated, and then they took leaves from that. So those are actually a generation of offsprings, if you will, from those parent trees. What Leslie tested on her leaf assay was the actual parent tree. So what I sent to Hannah, in theory, you would think that it would have 50%. So I thought that was kind of interesting. And so that's some more good stuff. And so like I said before too, we're going to be doing some of our own OXO testing. And it's one of those things that it takes more research to find the exact reason why the Ozark Chinquapin, um, it's unknown. It may be another series of genes, a set of genes, or another pathway. That's the unknown. But the Chinese chestnut and the Ozark Chinquapin, neither have the OXO gene in them. It was actually inserted to the transgenic American chestnut. And so right now we're coming up on time for break. And